Hello everyone, good evening. Today I will be talking about diffusion tensor imaging analysis. So I started learning about this with the help of my scholar mentor, and I have been learning about this for past few months. So diffusion tensor imaging actually is an imaging technique used in MRI, which is magnetic resonance imaging. So first, I will talk about a little bit about magnetic resonance imaging, and then I will move on to the DTI. So magnetic resonance imaging, MRI imaging, I think you have seen MRI machines, big machines in the hospital. It's actually a non-invasive imaging technique which uses strong magnetic field and radio waves to produce the image. We can use MRI to produce 2D, either 2D or 3D images. MRI is preferred over CT imaging because it's non-ionizing and it gives excellent soft tissue resolution and an excellent spatial resolution. And also, but there are many disadvantages as well. The machines are very huge and it's very costly. And if someone has metal implants in their body, they can't undergo MRI imaging. So, this is MRI machine. We have two types of machines, closed MRI machine and open MRI machine. So these open MRI machines, I actually haven't seen them in Sri Lanka, but they are used to take scans of people who have claustrophobia. Claustrophobia is they are afraid of enclosed spaces. So this open machine is used for them, and this is an example of MRI scans. So MRI imaging is mostly used for brain scans, but as you can see, the abdomen and the knee and the spine, there are also other body parts also can be scanned using MRI. And I will talk about MRI image acquisition briefly. So what happens here is, so there's a big magnetic field releases there's a giant coil inside the MRI machine, and then the H plus protons in our body gets aligned with this magnetization, and then from the MRI machine, an external RF pulse will be applied. Then these aligned protons get aligned in the transverse plane. So that's called transverse magnetization. And then after some time, this external RF pulse is removed. Then those transversely magnetized protons again get aligned into the current position. So this RF, so when they are aligning with the current position, they release some energy. So those releasing energy is detected by the MRI machine, and that signal is getting converted into the image. So there is this gradients applied because we are going to create a 3D image. So we need the information in X, Y, and Z axis. So we in the machine they use slide selection gradient. It's like a signal, and phase encoding gradient and frequency encoding gradient. And then if I go to the next slide, yes. So now I will be talking about the diffusion weighted imaging. So diffusion weighted imaging, just like I mentioned, is an MRI technique, But instead of these main three gradients, we are using an external diffusion gradient to get the diffusion information of the water molecules in our body. So that's how the diffusion weighted imaging is produced. So diffusion coefficient is it measures how easily water can move. So there are two types of diffusion: isotropic diffusion and anisotropic diffusion. So isotropic diffusion happens when water moves like freely in any direction, but anisotropic diffusion occurs when water moves in only a one direction. So that happens, mostly that happens in our nerve fibers, in our brain. So that's why we are using diffusion tensor imaging to analyze the white matter, the nerve tracts of our brain. So we can diagnose specific brain disorders like Parkinson and like other disorders, strokes, tumors. It's easier to diagnose those kind of disorders using diffusion weighted imaging. So so if I talk about diffusion tensor imaging, it focuses on the anisotropic diffusion. So that means the diffusion happens in the one side, only a one direction. So it takes advantage of the directional movement, and to like recognize the direction, which direction that the diffusion happens, it uses three methods: fractional anisotropy mapping, principal diffusion direction mapping, and the fiber tracking mapping. So diffusion tensor imaging is useful because it can help to map the brain's white matter pattern. So white matter, and there are two types of like matter, like matters in our brain. They are gray matter and white matter. So gray matter consists of cell bodies, and white matter consists of like the nerve parts, the nerve fibers. We call them axons. So axons carry all the electrical signals that carries the information throughout the body. So it's important to explore and examine the white matter tracts when it's come to like Parkinson and schizophrenia disease like that. And so, and also this fractional anisotropy values tells us how much the water diffusion is directional or asymmetric, and the fiber tracking map tells it traces the parts of the brain's nerve fibers. So in the DTI analysis that I'm doing, I'm using MRI-based. It's a Linux operating based software. So I had to learn Unix scripting as well. I'm not a pro at it, but I'm learning it still. And to use, there are many software like FSL and like AFNI. Yeah, there's another software called AFNI we can use. But MRTrix is very useful because it uses constrained spherical deconvolution to solve the like the fiber crossing crossing fiber problem. So crossing fiber problem is actually in diffusion MRI when two or more bundles of nerve fibers. So the nerve fibers, they don't go like parallelly, they cross at many places. So when two or more nerve fibers cross at that voxel in diffusion MRI image, we can't like specifically say that this is the voxel that these nerve fibers are crossing. So like then the signals get mixed and we can't get the correct information from that image. So we have to solve that problem. So using MR tricks, the method that they are using, constraint spherical deconvolution, actually does not solve the problem, but it reduces to a certain extent. I used a data set from openneuro.org. Actually, it's very difficult to find these MRI imaging data sets, and this is one that I could find. And if I talk about the warping effect, so the warping effect is the main issue. The physically visible in diffusion data. So that crossing fiber bundle is like internal problem. So physical problem is this warping artifacts. Warping artifacts is when we are doing the phase encoding to get the direction of that slice, to get the spatial information. We are doing encoding that using that spatial encoding gradient. So we are doing it like anterior to posterior means like front to back and back to front. That's what basically it means. So when we are doing the anterior to posterior, that side gets squished, like a magnetization force comes and that part of the images gets squished. And then posterior to anterior encoding is done to get the distortion data which happens in the AP encode. So that's what basically happens, and we are going to perform a pre-processing step to remove this effect. And so this is like the first we have to see the data and decide which pre-processing steps do I need to follow. Do I need to use denoising, or do I need to use that warping effect? We have to first look at the data. So this eval and bvec, that means so the B value controls actually how sensitive the scan is to water movement. That means when the B value is very high, we can get more diffusion data, more diffusion information about that image. So, but the downside is when the B value gets high, as you can see in this image, the resolution gets really low. To make the resolution high, we have to use a very large magnetization. That means in normal MRI, we use like 3 Tesla, but when we are increasing the B value, we have to use like more than like 30 or maybe more than that. Then we can increase the resolution as well. But in Sri Lanka, I think we don't have a machine like that. But in US, just like my mentor mentioned, she told me that John Hopkins University has these kind of machines, and they do the best research in this area. And so this is the 3D view, like the from the right, left, and the out. And then first we need to perform pre-processing steps on this data before we get the white matter tracts. First we have to reduce the random noise in the data, and then we have to use this Gibbs ringing artifact remover. So Gibbs artifact looks like ripples. So they don't we cannot see these Gibbs ringing artifacts in every image. So that's why looking at the image is very important before performing the pre-processing step. If we can see them, we can use the use the correct function to remove them. And next, we have reverse phase encoded images. So I mentioned that. So primary phase encoding is the main direction used for most images, and the reverse phase encoding captures the distortions introduced in the primary phase encoding. So we have to, what we are basically doing is we are just adding the B values and B vectors and then averaging them throughout the image. And next, if we need, we can so the famous disorder, and the most of the researchers have done in the Parkinson, for the Parkinson disorder. So for the Parkinson disorder, the whole brain doesn't, we don't need to analyze the whole brain. We just need to analyze only a region of the brain. So if we want to isolate that, only that region, we can use a mask. Otherwise, we don't have to. We can just, we don't have to use a mask. We can just avoid that step and just analyze the whole brain. And then this is the most important thing. So constrained spherical deconvolution. So in our brain, water moves differently in different parts. So I told you that we have white matter and gray matter. Other than that, we have what is it? Cerebrospinal fluid. So cerebrospinal fluid is a fluid which actually protects our brain and the spinal cord of our body. It's a liquid. So white matter, it moves along the fibers in one direction. But gray matter and cerebrospinal fluid, it moves in all directions, like anisotropic and isotropic diffusion. So to remove that crossing fiber problem, we are using this method. By using this method, we can actually map the directions, and we can get the directions of the white matter tracks and the where it crosses. So what this does is, first we create a template for how water moves in white matter, gray matter, and cerebrospinal fluid using the person's own brain scan. And actually, this is very important because then we can analyze each person uniquely. And then the scan data is split into small region. And then for each voxel, we figure out which direction what is moving. And if there are multiple fiber directions, crossing that spot. And so this is the resultant image. So the green color represent the gray matter, and the red color, there are only like few, represent the cerebrospinal fluid, and the white matter representing blue. So actually CSD, which means constrained spherical deconvolution, improves fiber orientation distribution, which is essential to analyze the pathways of multiple fibers, because otherwise we do not know where the fibers are crossing, and we do not know which direction should we go after that certain crossing thing happens. The next step of this analysis is creating tissue boundaries. So creating tissue boundaries, we do this to separate the gray and white matter, because only the white matter matters for us. White matter only have the nerve fibers, and actually this is used to place seeds for tracking neural pathways. And what we do is we use the MR tricks to segment the brain's anatomical image into 5 type of tissues. So gray matter, subcortical gray matter, white matter, cerebrospinal fluid, and pathological tissues. So those 5 are visualized in here. So there are only 4, but in that tutorial, they have mentioned that if they cannot visualize it correctly, it doesn't appear here. So that must have happened here. And then we align the anatomical image with the diffusion weighted image, which we performed CSV to ensure the accurate tracking results. And then finally, we create a boundary. And this red color is the red color line thingy is the boundary that we create. And next part in our analysis is to create a streamline. So streamlines represent connections between white matter regions. So white matter and gray matter regions. For that, MRTrix uses a probabilistic method to create many streamlines for each voxel. So voxel is actually the volumetric representation of pixels. So in pixels, we have height and length, but in voxels, we have the depth as well. So actually, this process is after generating this streamline, this is called anatomically constrained tractography. Tractography because we are creating tracts along the gray matter and white matter. Actually, we do not need every streamline going through the from gray matter to white matter. We only need the that tracts inside the brain because we are only considering inside the brain. And so the next and the final step is creating the connectome. So connectome is a map that shows how different parts of the brain are connected by the streamline. This is very important to understand how different regions of the brain communicate and work together. So since now we have created all the tracts in our 2D images, now we have to connect them across the 3D volume. So that's why we use the connectome. So to create the connectome, we first need to divide the brain into different regions using the atlas. So atlas means, I think you guys have heard about the atlas of the world. It's the map of the world. So the atlas of the brain is also the map of the brain. So there are many atlases which have founded by many researchers. So in this tutorial and this analysis, I'm using free surfer atlas. So this Desikan, Kiliani, and I don't know how to pronounce that. So those are the scientists who have found this. After researching about the brain, they have created these atlases, and for each color, you can see there's a separate region. So that's what actually this does is. So after creating a connectome, actually this is a correlation matrix. So you can see that there are 2 distinct boxes. Those 2 distinct boxes are for left and right hemisphere. And these bright color yellow spots are the regions that are connected together. So we have this X and Y axis. So X and Y axis have, each of these boxes have a number. So there's a lookup table. Just like this lookup table, we have a lookup table. We have a lookup table for this connectome as well. If we want to know the region of this this yellow color spot, what we do is we get the location, and then we look from that lookup table. So what's the region which is connected to? Then we can use this information as a feature in our machine learning model as well to analyze a group of people. And that's actually all about the DTI analysis. And after performing all these steps, we can, this is only for one person, but just assume that we have like a group of 100 people. So it's very difficult to perform this step one by one, because some steps took, since I don't have a very powerful laptop, some steps took like more than 5 hours to complete. So if we have like 100 people, a group of 100 people to perform these steps, we can use scripting to automate everything. And then we can use these results to create a machine learning model, or to do some like comparisons with the other MRI imaging, other diffusion tensor imaging analysis software to use in our project. So these are some future works that came top of my mind, which can be used, this DTI analysis. So yes, that's all from my side.